Okay, so here's the splash page, and we're going to enter in our username and then go to the hemocytometer page where we'll create a new spread and start counting. And the thing that will help us count is clicking on the little yellow things, and then we can put a number into quadrant A. We'll do the same for quadrant B. Again, this clicking and being able to mark the cells with the yellow dye is a very useful way of determining what you've done and what you haven't done. We can zoom in and scan around, which is very, very useful, so we can actually look to see which balls are on the line and get a better view of things. So we'll do that with quadrant C now, flicking through and marking all the cells that we want to count with yellow. And these, of course, are the live cells. The blue cells are the dead ones. They still need to be counted, and we'll do the same with quadrant D. Now, after we've done that, we can get the computer to tell us what the results were. And we might find that there's a discrepancy, so we might want to go in and have a closer look. So if we go into quadrant D and just zoom in a little bit at the goal line and see where we might have gone wrong, because you can see that the computer is actually flagged with an X1 there that looks like it's well over the line. So we're really wondering what's happening there. And of course the key thing is if you start to pan and get that quadrant right in the middle of the field of view, then suddenly you can see that one in the bottom left-hand corner is just touching the left line. Same for the ones in the top. Now, you can do dilute samples. If you've got a sample that's too dilute, you can concentrate it. You can go the other way and make a sample more dilute if it's too concentrated. You can repeat one as well. So lots of different options there. Everything sent to the server can be checked against the server for assessment purposes. Let's have a look at the Plaque Assay one now, generating a new Plaque Assay. Lots of plaques to count. Again, little counting aid, clicking on them, zooming in. This time we've got a little active counter in the top right-hand corner of each well to help us keep count. We can submit our values to the server, and they, of course, can be checked for assessment purposes. Not particularly exciting. The agglutination one is quite fun. Loads and loads of plates can be generated, and you can generate an almost infinite number. You can zoom in and out. You can set the assay up and just run it and get tons and tons and tons of data. And that's basically it. So thanks very much for watching.